we are going to discuss on the inverse fluidization and its application for the treatment of the wastewater. Now, as all of us know that our earth surface is covered by 71% water, but only 3% fresh water is available to all of us. So, here as 3% only fresh water available to us, and it is also second things which is most required after the air for all living organisms or living beings on the earth. And the water which we after uses we generate, that is means in the different sectors, there may be domestic sector, commercial, agricultural, or the transport sector. So, whatever after use are generated, that is the wastewater, and excluding the domestic sector, the water generated in other sectors are known as the wastewater or the industrial wastewater. So, we can call in the wastewater which is generated in the domestic sector as the municipal wastewater, and in other sectors that is the industrial wastewater. As these waters, industrial wastewaters contain various toxic and hazardous pollutants, and they contaminate with the surface water as well as the underground water. So, that is why our 3% fresh water also getting contaminated due to our day to day activities and due to the industrial wastewater being that is discharged to the ground without being treated. So, thus it is required to treat them before discharging, and this treatment should be proper, by which the toxic or the hazardous pollutants, it may be some organic, inorganic substrates also, it should not be mixed with the water bodies. Now, otherwise there will be health issues will be there, shrink in the food productions, and the it will environmental issues also will be there. So, here when we are going to treat this wastewater before discharging into the main water bodies, what parameters we should consider during the treatment of this wastewater. The parameters such as BOD, biological oxygen demand, chemical oxygen demand, total organic carbon, and certain metals or the microorganisms, inorganic nutrients, it may be nitrogen, phosphorus, sulphate and many more. These factors should be considered during the treatment of the wastewaters, and depending on the type of the pollutants, different methods are being used to treat this industrial wastewater. Typically, wastewater treatment methods are categorized in three levels, that is primary and secondary and tertiary types, which consist of the different physicochemical and biological methods. Now, secondary and tertiary methods are the known as the advanced methods, and example absorption, adsorption, ion exchange, membrane based separation or the biological processes, where the primary method, primary category is the conventional methods will be used. Now, among these biological unit processes is gaining popularity since early 80s, and where we adopt the technologies to remove these suspended solids and inorganic, organic sulfates, ions, and also soluble and colloidal organic matters from the industrial wastewater. And these processes use a great variety of the reactors, including batch reactor, complete mixed reactor, plug flow reactor, packed bed reactor, fluidized bed reactor, etc. Now, the fluidized bed biofilm reactor, which may be aerobic or anaerobic type, are also used under this secondary type of the treatment. Among these reactors, this fluidized bed biofilm reactors, that is aerobic biofilm reactors, use the fluidized bed technology, and that is being currently used or in the recently being in the use for the treatment of the wastewater. Now, this aerobic fluidized bed biofilm reactors is known as the inverse fluidized bed reactor, and here the industrial wastewater containing the different pollutants are being handled and treated, by which this pollutant level is decreased to a great extent, which we will discuss with some case studies. We will discuss in this 1 hour lecture. Here normal 3 phase fluidization systems, the liquid is usually taken as the continuous phase, where the it may be the co-current or the counter current. Usually the liquid which is the continuous phase, it flows in the upward directions, co-current upward flow of the gas and the liquid, and here the particles are usually denser than the fluid being used, whether it is the gas or the liquid, and the upward flow of the gas and liquid which counter the net gravitational force of the particles with which the fluidization takes place. But when the density of the particle becomes smaller than the density of the liquid, the fluidization will be carried out in the downward direction, because when the lighter particles are used, the movement of the particles will be in the downward directions, liquid flow downward directions, which is in the counter current to the upward flow of the gas. Gas is supplied from the bottom, liquid is supplied from the top, and the lighter particles initially remain on the top level, but with the increase in the flow rate, the solid particles move in the or fluidize in the downward direction. This process will be known as the inverse fluidization. Here you can see the first figure, it is shows the static bed, that is when the inverse fluidized bed, the liquid is the that is column is filled with the liquid and some solid particles which is lighter than the density of the liquid. The first figure shows the static bed, that is it is not fluidized in this case, just bed materials forming a packed bed, all the particles remaining on the top of the bed, as it is as all the bed materials are lighter than the water or the liquid. And the second case, you can see the video how the materials getting fluidized. Here you can see when we increase the flow rate of the air, because water flow rate is maintained adjusted to maintain the steady state, because whatever the water will be flowing through the inlet, that maintaining the level of the water in the column, some water will be discharged through the outlet. So, inlet and outlet flow rate are adjusted to maintain the steady state in the column. Now, by increasing the velocity, how you can see in the this second case, first case was the static bed, second case the materials was fluidized, but in the lower part you can see the some space is there, that is a thin layer of the particles are found in the lower part, but in this video where the second part, it is almost covered, all the particles covering the entire section, entire height of the column, the particles are entire height of the column is covered with the particles, or the particles are covering the entire height. You can, that means particles are getting fluidized all along the length of the column. And if you last figure, if increasing further the velocity of the fluid, that is the gas, we can see, because water flow rate here we are not varying, just here more vigorously, as velocity we are increasing, more vigorously particles are getting fluidized. Just this is just a short video for the fluidization of the particles in the downward directions with the increase of the velocity. Now, let us move to the our slides which will. So, just in this, as gas flow upwards, the solid particle and this gas which flows upward, this exerts some pressure on the particles and water flows downward. So, because of this force, net force on the particles, particles tend to move downward, and again because of the tendency of the lighter particles to move upward again, the particle also moves again with once again when it is subjected to the force exerted by the buoyancy force by the water and net force of the water and the air, particles float down also. So, thus there is always movement of the particles in the upward and downward directions. Here we can see just a schematic diagram of the co-current and the counter current inverse fluidized beds. Here we can see in the first figure, which is on the left side, counter current means the water is flowing from the top, allowed to flow from the top, gas is allowed to flow from the bottom. Here you can see initially before starting the increasing the velocity, all the particles remain on the top level, then gradually when the velocity is increased, then the flow rate of the water is increased and the gas is also increased, the particles tend to move downwards. This is counter current because movement of the water and the gas is there. In the second figure, that is co-current flow, here gas and liquid all are supplied from the bottom, and liquid flows out collected or the outlet is from the side of in the top region, and gas outlet is from the top of the column. Here also the accordingly the flow rate is adjusted, so that the at what rate we need the fluidization, because for the biological action the vigorous movement is not desirable. So, for depending on the type of the applications, we may have the vigorous movement or the smooth movement of the solid particles in the bed. So, here we can see that there are many advantages for this inverse fluidized bed bioreactor, which we use for the treatment of the industrial wastewater in terms of the BOD, COD, total organic carbons, and also that is removal of the ammonia, nitrogen, phosphate, sulphate, phenol or other toxic pollutants, that is like hexavalent chromium and that is phenol etc., can be removed using the inverse or can be degraded using inverse fluidized bed bioreactor. It has advantages that such reactors are compact, efficient with a small floor area requirement and the low energy consumption also, and higher biofilm surface area is provided with the bed materials, and high mass transfer rate and higher biomass age also, as smooth fluidization is maintained. So, here biomass and also with the continuous supply of the wastewater, the biomass gets the feed continuously, because of which the biomass age also higher in comparison with the other bioreactors, as well as these reactors are also resistant to the toxic loads and the temperature variations. As it is wastewater is taken, so sometimes during the operations sudden temperature rise if occurs in other bioreactors, there the microbes may die, but here as it is wastewater is taken considered as the main medium. So, here if sudden rise in the temperature is there, that is taken care of by this water, wastewater. As a result, the overall temperature does not rise suddenly, and where the microorganisms, that life of the microorganisms is not affected. These reactors are also used to have larger degradation rate, higher organic loads uptake capacity, means high level of the high concentration of the pollutants also can be degraded, and where degradation rate is also high, and construction cost also very less and do not show any clogging problems. As you from the video you saw, there is no complex arrangement by which there is no clogging problems also, whereas other reactors the clogging problems will be there. Applications, it is mainly used to treat the wastewater, industrial wastewater, collected from the different industries such as brewery, starch, textile, sugar, steel, dairy industries, also various other wastewater such as municipal wastewater, which contains the inorganic, organic pollutants, and harmful industrial pollutants like aniline, phenol, sulphate, ferrous iron, ammonia also they can be degraded using these inverse fluidized bed bioreactors. And dimensions, the reactors with varied dimension configurations also have been used under different operating conditions. Depending on the quantity of the wastewater to be handled, the dimensions can be varied also. And treatment removal efficiency also found to be dependent on the hydrodynamic features of these inverse fluidized bed bioreactors, and what type of the microorganisms we are using, and nutrients present in the wastewater, that is pollutant present in the wastewater, and concentration of the level of the pollutants present in the wastewater. So, accordingly reaction conditions can be maintained, that is the pH level. Reaction conditions mainly refers to the pH, what pH we have to maintain such that the microorganisms will live longer. Now, wastewater categories, as you told that industrial wastewater, whatever the water generated after uses in different sectors excluding the domestic sector, that is the industrial wastewater. And if we consider the domestic sector, that is municipal wastewater, so all are the wastewaters, that is water generated after uses. So, this industrial wastewater can be categorized in 3 groups, that is physical, chemical, biological, 4 groups, that is physical, chemical, biological, and radiology, depending on the characteristics, which may be physical, chemical, biological or radiological characteristics. So, physical characteristics include the total solids, that is volatile, fixed, suspended, dissolved, and settleable solids, color, odor, that is physical characteristics, the solids having the physical characteristics, they are referred to as the wastewater of the physical in nature, which are these physical characteristics are to be handled or the treated. The chemical characteristics, where the various organic and inorganic components will be present, and organic constituents comprise carbohydrates, fats, oil, grease, pesticides, phenols, proteins, surfactants, and some volatile organic compounds also. And inorganic constituents may be alkalinity, chlorides, heavy metals, nitrogen, phosphorus, pH, sulfur, and some other toxic compounds, whereas biological features include various microorganisms, which may be aquatic plant or animal based, and aquatic bacteria and viruses. Some of the microorganisms may be pathogenic, which is also harmful in nature, while some degrade the organic content of the wastewater also. And the sources of the radioactive, that is radiological characteristics, that is because of the radioactive wastes, which these radioactive waste include the mining and of the radioactive materials and those generated from the nuclear power plants also. So, depending on the waste generated from the different sectors, these industrial waste categorized in these 4 categories. Further, here the US Environmental Protection Agency also they just categorized this wastewater into mainly 3 categories, that is conventional, non-conventional and priority types. Whereas conventional types, they treat the conventional pollutants like BOD, total suspended solids, pH, oils. Oils may be from the mineral sources, animal sources or the vegetable sources. And non-conventional pollutants, which include total organic carbon, COD, fluoride, hexavalent chromium, ammonia, nitrate, the phosphorus, manganese, phenols and sulphate, etc. There are also 129 numbers of the priority pollutants which are reported in literature. So, which are so vast we are not discussing. So, just to have the idea, as per the US Protection Agency, that is there are 3 types of the that is wastewaters are there. Now, coming to the just literature, so in short we will see literature, then we will go to the case studies. Here we can just see that when the density of the particles is less or the close to the that of the liquid, the inverse fluidization is possible, and it is because of the upward gas flow and the mainly the counter current inverse fluidized bed is used for the treatment of the wastewater. And here again for this for proper fluidization to be carried out, we need to maintain the particular velocity, means we need to know the proper velocity at which properly fluidization will be carried out, and also gas hold up, phase hold up, etc. So, this when the fluidization is smoothly carried out and all the particles are fluidized, and that particular velocity will be referred as the minimum fluidization velocity. Separately one can know that what is the minimum fluidization for the gas and what is the minimum fluidization for velocity for the liquid or the water also. And we may have the turbulent fluidization also, but depending on the applications. For the bioreactor, when we are will be using microbes after culturing the microbes, then for that we need the smooth fluidization, not the turbulent fluidization. So, initially when we start increasing the flow rate, as we saw in the video, the entire bed that is the concentration of the solids will not be uniform along the entire axis of the bed. High concentration of the solid will be there near that that is liquid distributor, that is in the top of the bed, and with the further increase in the velocity, solid hold up will be uniform throughout the bed, as in the video we saw. Now, the successful design of these gas liquid solid fluidized bed systems depend on the accuracy prediction and the fundamental properties of the systems. Now, as we know that this accurate prediction of these fundamental properties, such as the gas hold up, pressure drop across the bed, minimum fluidization velocity, bubble size, bubble rise velocity, friction factor, voidage, mass transfer coefficient are some of the important factors. But here for the inverse fluidized bed, we will be considering few factors, that is in addition to these phase hold ups, also we need to know what is the that is aspect ratio and settled bed volume to the reactor volume ratio, all the also, and retention time also we will be discussing also. And both here inverse fluidization can be made easier by small density difference between the fluid and the particles. So, there as per the researchers, we can researchers predictions, Benedict et al. They developed the HF by H0, that is expanded up to what height bed is getting expanded, and H0 is the static bed or just before fluidization, that is this expansion, you can say bed expansion ratio was expressed by the researchers in terms of the Reynolds number and the Archimedes number. So, here other researchers also carried out, mainly Pan and Pan et al. In 1982 suggested 3 type of the models to determine the that is phase holdups, and also to know that how they are dependent on the gas velocity and liquid velocity. So, different researchers also further worked on this, and they also added to some modified some these equations and also or correlations, and some of these are just we are summarizing. Detail these correlations we are not discussing because it will take time. We will just case studies we will be discussing. Here the as I told that is main is the phase hold up, where the liquid hold up, gas hold up and the solid hold up also we need to know, and also hydraulic retention time, biomass support particles, what type of the support particles we are considering, and bed aspect ratio, superficial gas velocity, liquid velocity, bed expansion, and the settled bed volume to the reactor volume ratio are the important parameters which are to be studied for the for knowing the hydrodynamics of the inverse fluidized bed reactors, the bioreactor. Here for the phase holdups in general it was observed that if the because this phase hold up they important role play important role to know the effectiveness or to decide the effectiveness of the inverse fluidized bed bioreactor. It is observed that when we are increasing the gas velocity or the increasing the liquid viscosity or the particle density, how the phase holdups are changing. Here it is observed by the various researchers that this phase hold up, gas phase hold up, phase hold up means fraction of the gas in the bed, that is the gas phase, how much gas is present in the bed. It is observed that this gas phase hold up, sorry it was epsilon g, it is why it is important, because it is crucial for the transfer of oxygen from the gas or the air to the liquid phase. When because for the growth of the microbes, then as aerobic bioreactor, growth of the microbe, oxygen is required. So, this gas hold up helps or the makes it possible to transfer, to supply the oxygen to the microbes. So, here oxygen level in the liquid phase is essential for the effective growth of the micro organisms, and these researchers have found that this gas phase hold up increases with increasing the gas velocity. It is obvious, if more gas is supplied to the bed, naturally fraction of the gas in the bed also will increase. So, gas hold up it was increased with the increase in the gas velocity, and it was also observed that if you increase the superficial velocity, that has negligible impact on the solid hold up, that is naturally the gas hold up. Gas because if you increase the liquid velocity means more liquid will be there. So, that has no impact on the gas hold up, and here also gas hold up is reported to increase with the increase in the while in the slugging fluidized bed regime. If it is not properly fluidized, slug formation is there, that is layer wise formation is there in the bed. In that case, it is this gas hold up may increase with the increasing liquid velocity. Otherwise, in the normal the fluidization regime, the gas hold up is not dependent on the liquid velocity. So, also it is observed that liquid hold up increase slowly from the bed to the freeboard region over a wide transition region. Naturally, if the liquid velocity is increases, the liquid hold up will also liquid hold up, that is fraction of the liquid in the bed will be increasing, and it is observed that in the freeboard region, naturally in the bed region more solids will be there. Freeboard means after the fluidization regime, the region is the freeboard region, there more liquid will be there. So, liquid hold up is more in the freeboard regions. So, researchers have worked on the by taking the polyethylene particles or polypropylene particles and they studied the this on the phase hold up, and they found that this liquid phase hold up increases with increasing gas velocity as well as the liquid velocity, whereas at higher liquid velocity this hold ups value remains nearly constant, because they maintain the, when the fluidization will be at the, as in the last video we saw that it is already the increasing, and it is covering the particles covering the entire length of the column. If you further increase the gas or the liquid, naturally liquid hold up will not increase, it is maintained, steady state we are maintaining. So, further increase in the up to certain level liquid hold up will increase, then after that the liquid hold up will remain constant, because as it is the level of the water in the reactor is maintained, steady state is maintained. So, here liquid hold up decreases with increasing the gas velocity, and liquid hold up also increase with the, it is the normal fluidization regime, but after certain level it is liquid hold up will not change. So, the solid hold up similarly decreased from the bed region to the freeboard region. Naturally, bed region more in the top, as you in the figure we saw, more solids are there in the top region, that is the bed region. So, there solid fraction is more, so solid hold up is more. In the freeboard region towards the lower part of the bed, it is the, that is the freeboard region, there less solids is there. So, solid hold up is less in the freeboard region, and the solid hold up is found to decrease with the increasing gas velocity. Means when more gas is supplied, the it is fairly, means what happens, it is the means thin layer of the solids will be there in the freeboard regions. So, the solid hold up decreases. Similarly, it is observed that solid hold up decrease with increasing the gas velocity, liquid velocity as well as the viscosity of the liquid also. If liquid becomes thick also, solid movement will be hindered, or the getting resistance to move freely into the bottom regions. So, these are the some correlations are proposed by the different researchers who studied on the bed hydrodynamics, that is gas hold up you can see here, all containing the terms, that is gas velocity, liquid velocity, liquid viscosity, and on the liquid phase hold up also. So, it is so considering the that is gas velocity, liquid velocity, density of the gas, liquid, and solid hold up, which is the mass of the solid divided by, here you can see it is the volume of the reactor, means mass of the solid by the density of the solid, that means it is the volume of the solid divided by the volume of the reactor, that is the how much fraction of the solid is there within the reactor. So, solid hold up. So, total sum of the all the hold up should be 1. So, these are the various correlations suggested or developed by the different researchers. Now, minimum fluidization velocity, as already we told, that is also we have to means during the experiments on the minimum fluidization in the using the inverse fluidized bed bioreactor, minimum fluidization velocity for both gas and the liquid has to be determined, because under the minimum fluidization condition only all the particles will be fluidized. Before that it will be partially packed and partially fluidized. So, for which minimum fluidization velocity is to be maintained for the smooth fluidization, or it can be slightly more than the minimum fluidization velocity. And so, different researchers also worked on this and predicted considering the different solid particles, different materials using, and they proposed the correlations for the minimum fluidization velocity also. So, these are the minimum fluidization for the liquid fluidization velocity, and for the minimum gas fluidization velocity. So, in terms of the here gas fluidization velocity, which was determined in terms of the Reynolds number, viscosity of the gas, Archimedes number. So, different researchers have worked on this. Here also you can see the liquid minimum fluidization velocity, Reynolds number, viscosity of the liquid, and some Archimedes number. So, have been used to predict the minimum fluidization velocity. Then hydraulic retention time is also another important parameter for the knowing that how much time is required for degradation of the pollutants from the wastewater. So, it is the hydraulic retention time is calculated, that is T equal to V by Q, volume of the reactor, Q is the wastewater flow rate in the reactor. So, T is the hydraulic retention time, that is how much duration wastewater remained in the reactor for the treatment. So, knowing this, we can it is observed that inverse fluidized bed bioreactor, the retention time is less in comparison with the other reactors, because here this aerobic treatment with the low concentration of the which is easily degrade the low concentration of the pollutants, for which oxygen requirement is less. That is why with the low retention time also degradation becomes that is effectively done. Now, it is observed that percentage of the COD reduction also increases, that is in the inverse fluidized bed reactor with increasing the hydraulic retention for the domestic wastewater and the starch wastewater. It is the some case study, it was for the, although the low level of the pollutants can be degraded with less time, low retention time, but it is found that for the COD reductions in comparison to the that is for COD reduction from the domestic wastewater and starch wastewater, slightly higher retention time is required. So, this also depends on the hydrodynamics and the nature of the microorganisms used. What type of the microorganisms we are using, on depending on that we can decide how much retention time to be provided. So, low density plastic biomass support particles, here in the inverse fluidized bed, as in the figure we saw, there that is orange colored polypropylene beads were being used. So, here low density support particles, they provide the surface for the growth of the microorganisms. So, this that as they provide the support. So, low density biomass support particles are the also important part in the inverse fluidized bed reactor, because when they grow, they attach to this surface, then they take the pollutants from the wastewater as the feed. However, the attachment to the microorganisms also on the solid particles depends on the surface roughness and the degree of hydrophilicity. So, accordingly researchers worked considering the different materials. They observed that if the material is hydrophilic and also surface is rough, then more microorganisms can attach to the surface. And for that, some researchers they taking the other materials, that is polyvinyl chloride, they treated the these PVC particles with the solution of the chlorosulfonic acid and also polyethylenine to improve the biofilm formation. So, if you are not taking the hydrophilic material, then or hydrophilic surface, then we have to treat these materials before using in the biofilm reactors. So, the intention is to have the that is hydrophilic nature and the rough surface of the particles, and but usually the spherical particles or the approximately spherical particles are taken, because irregular particles affect the fluidization process, and if you use these irregular particles, then bed stratification may take place. Stratification means layer wise formation may occur. So, to avoid that and to have the uniform growth on the surface of the particles, to have the uniform fluidization, usually spherical particles are taken or closely spherical particles are taken, such that smooth fluidization will be there and the growth also will be proper in the inverse fluidized bed biofilm reactor. Now, bed aspect ratio also another important factor. and bed aspect ratio that is h by d, height of the bed to the column diameter. When we are taking the column, diameter is fixed. Suppose we have fabricated one column, diameter is fixed, we cannot change the diameter. Only the bed aspect ratio can be varied. If we bed height, we vary. Bed height means material we can vary, then aspect ratio can be varied. But here with rise in the when we are changing the bed height, that means we are taking more and more particles. As a result, that will require more gas velocity for the fluidization. So, that is why it is observed that with more particles, large number of the solid particles will require more because more surface we are providing, and that means more air flow will also be required. First thing, that is for means for fluidization, when the more materials you are taking, more air will be required. Second thing, more materials means more microbes, microbes getting more surface. So, number will be multiplied frequently, for which they will needing the microbes will be requiring the more oxygen also to grow. So, reactors having low aspect ratio require the low UG, that is gaseous velocity. It is obvious, less material means less air velocity will be able to fluidize the materials. Again, aspect ratio of the, it is observed that by the researchers that if aspect ratio of the 10 is maintained, then petroleum and brewery wastewater can be treated for the COD reduction up to 64%, and phenolic wastewater, it is observed that degradation rate decreases if the bed aspect ratio is increased above 1.787. That means, it is the optimum level of the bed aspect ratio for the phenolic wastewater. So, that way with the researchers we can if we want to carry out the experiment or we want to treat the wastewater for the COD reduction, then accordingly we can just getting the information, we can close to that value, we can maintain the bed aspect ratio such that more effectively COD reduction can be carried out. Now, superficial velocity also another important aspect. So, COD removal is found to increase with increasing the gas velocity. Naturally, more oxygen will be supplied. So, but still if it is not that we go on increasing, then it will be giving more oxygen for the growth, but there is some limit, that is optimal value of the gas supply should be there. After that the COD removal will be decreased. Sometimes more aeration, but there is more oxygen is there, but there is no pollutants in there, or other conditions will be affected, pH will be affected, or the viscosity will be affected, and the fluidization quality, minimum fluidization quality means fluidization will be converted from the smooth to turbulent also, because of which the biofilms which are already formed on the surface of the particles may get damaged also. So, that is why there is some optimal limit. So, beyond that it is decreases. Similarly, gas hold up is found to increase with the gas velocity, while the biodegradation also observed to decrease after the optimal value. So, although up to certain limit there is the means gas hold up or COD reduction increases with the increasing gas velocity, but always there is some limit beyond which it will not it will have the negative impact. So, with increasing the liquid velocity also fluidized bed happen to expand, naturally expansion takes place, but here it is observed initially that liquid hold up increases and solid hold up decreases, bubble size, because solid hold up decreases, that means bubble size will reduce, gas solid interfacial will also be, gas liquid interfacial will increase, and here the minimum fluidization velocity for the gas will be decreasing, and volumetric mass transfer coefficient will increase. So, these are the findings of the researchers based on their research works using different materials, different systems. Another important parameter is the VB by VR ratio, that is settled bed volume. to the settled bed volume means after fluidization if you shut off the air supply, then the what is the bed volume, that is the settled bed volume to the reactor volume. So, here reactor volume means entire fluidized bed volume. So, important parameter. So, here for any given mass of the bed BP subscript is the bed particles, and minimum fluidization velocity for the gas is found to increase with the increase of the Vb by Vr ratio. And for any mass of the bed particles and the superficial velocity, gas hold up is found to increase with the increasing the this Vb by Vr ratio also till the optimal value is reached. And also sometimes it was observed that optimal value of the Vb by Vr decrease with increase of the mass of the solid. Naturally, this aspect ratio Vb by Vr ratio will be changing with the, because settled volume, if we are changing the mass of the bed, naturally volume of the bed also will change. As a result, Vb by Vr ratio will be changing also. So, here COD reduction is found to decrease with rise in the oxygen flow. If more oxygen is supplied, that because this microorganisms may die also because of the turbulent fluidization, as a result COD reduction will also be affected. So, these are the some correlations suggested by the different researchers for the knowing the Reynolds number, minimum fluidization velocity for hydrodynamic studies, where the two phase systems they determine the pressure gradient also. So, these are the few expressions which are suggested for the gas hold up, minimum fluidization velocity, Reynolds number, bubble velocity. So, now we will go to the experimental part for the treatment of the industrial effluent. So, hydrodynamic study helps us to know the conditions of the bed to be maintained to have the smooth fluidization, and with which we can be able to treat the wastewater properly without affecting the life of the microbes. So, here in the inverse fluidized bed reactor, which is was considered as the least important in the past, but now it is gaining importance owing to the various industrial applications, mainly for the wastewater treatment, because it removes the contaminants and the organic material using the less energy and the low cost involvement also. Here several physical, chemical, biological methods are there, as already we discussed. Here it is found that higher efficiency on the removal of the pollutant is observed in the inverse fluidized bed reactors, and other various advantages also is there, as already we discussed in the advantages part. So, it is recommended for the treatment of the wastewater, and here various industrial wastewater case studies are there, and in our lab also we have already studied the some sugar industry wastewater, textile industry wastewater, steel industry wastewater, and some other inorganic such as phosphate, sulphate removals also we have studied. And so, using these three phase inverse fluidized bed bioreactors, we can say that we can successfully treat the industrial wastewater and also municipal wastewaters. Here this three phase inverse fluidized bed reactor has inherent advantage over the two phase systems due to the improved supply of the oxygen and the elimination of the pre-aeration, because sometimes to supply the oxygen to the microbes, if you are taking the two phase systems, so then initially we have to supply some oxygen, aerate the system, so that microbes can survived. But here in this 3 phase we will be continuously supplying the air for the aerations also. So, steady biomass loading is required, so which is also possible in the inverse fluidized bed reactor easily, and it can be controlled in the inverse fluidized bed bioreactor, where the light particles are fluidized to by the downflow of the liquid, because water is supplied from the top. And here we will discuss some phenol, ammoniacal nitrogen, phosphate ion, sulphate ion from the research works which you have already carried out in our lab. Now, chemical oxygen demand, which is calculated, as we know, this is one of the important parameter for the treating the wastewater. So, it is an indirect measure of the amount of the organic compounds present in water. It is often used as a rough approximation of the pollution level of concerned wastewater. So, we can calculate using this formula A minus B into N into 1000 into 8 by V 0. Here A is the ml of the ferrous ammonium sulphate used for the blank one, B is the milliliter of the ferrous ammonium sulphate used for the original sample, and N is the normality of the ferrous ammonium sulphate, and V naught is the ml of the sample, and 8 is taken because it is the atomic weight of the that is nascent oxygen we are taking. So, thus COD you can calculate. Similarly, BOD also measures the oxygen which is utilized by microorganisms under the specified experimental conditions during biological oxidation of the organic matter which contained in the liquid waste. So, this is the just schematic diagram for the experimental setup which is there in the lab. This is the inverse fluidized bed reactor. Here the you can see that here the through this rotameter, that is the air is supplied and water is supplied from the top, which is again recycled back, means collected in a drum, and it is again supplied to the column from the top. Here manometers are connected. If we need to know the measure the pressure drop at a different levels, so different manometers we can connect here also. So, it is a perspex column and these are the bed materials, and here on the top one mesh or screen we can provide to prevent the upward, if you suddenly if you increase the water flow rate, then the particles may carry over, means water may overflow the column. So, to prevent that, some screen we may we need to provide on the top of the solid particles here in the or the top layer of the bed. Here just picture of the experimental setup, inverse fluidization. Here the procedure how it is carried out, as two flow is there, liquid flow and the gaseous flow, two rotameters will be there. So, that can be calibrated, and then the before taking the experiments or carrying out the wastewater treatment, and the column can be loaded with the solid particles of the required size, and water usually in as per the research it should be within just 2 to 10 centimeter size is taken, whatever the data is available in the literature. Here in our lab we have taken 6 mm size particles. So, water is used in the fed from the top and air is supplied from the bottom, and water flow rate is attained means allowed to attain the steady state by adjusting the inlet and the outlet flow rate, and manometers will be connected. Water flow increases gradually in steps till the bed is completely fluidized, and initially it will be packed bed, as in the figure video also we saw. Initially bed is the packed bed, then partially it is fluidized, then it is smooth fluidization occurs. So, we have to maintain that smooth fluidization corresponding to minimum fluidization velocity. The pressure drop plot also we can from the by plotting the pressure drop versus velocity, we can get the minimum fluidization velocity also. And here then once the minimum fluidization is attained or the smooth fluidization is attained in the bed, we can introduce the cultured microbe which is separately cultured inside the column and allow the microbes to grow within the column under the steady state conditions and the minimum fluidization conditions. Then after few weeks, when it will be the microbes will be 2 to 3 weeks time, we can collect the samples, so that microbes will be grown, and then we can verify that whether the films have been formed or not using the technology. Here usually in our research that is work, the students had used the eSEM to measure the biofilm formation, thickness of the biofilm formation, and that again indirectly also concentration of the sample wastewater which is there in the column can be collected from time to time at regular intervals, 6 hours, 10 hours intervals, and to check that whether what is the level of the pollutants in the sample, so that we can know that it is getting the pollutant level is getting degraded gradually or not. Now for the phenol degradation, the inoculum was activated sludge was the activated sludge taken from the biological unit of the steel plant, local steel industry, and there some it was wastewater was enriched with the mineral salt. Then the that is taking the it is cultured, and then it was introduced in the inverse fluidized bed reactor. Similarly, for the sugar industry also, the inoculums were collected from the sludge taken from the starch industry, and inoculums was also introduced, and another was some experiment, I do not remember exactly. We had collected the wastewater for that inoculum was the wastewater of the dairy, that is unfed wastewater we had collected to prepare this, to culture this microbes also. So then here if the difference in the weight of the biomass in the reactor at 2 consecutive samplings differs only within 5%, it is considered as constant biomass growth. If that is suppose 6 hours interval we are collecting the samples, we can check that what is the that is weight of the biomass. So what was the biomass in the first sample, and what is the biomass in the second sample. If there is the 5% difference, that means growth of the biomass is there. So we can ensure that biomass being grown in the reactors. So biomass slugged up from the particles and separated from the effluent in removed from the system to determine the COD reduction experimentally as per the equation. COD or the whatever the pollutants will be there. So just this is the general procedure, whatever may be the pollutant, this is general procedure. Now it is for the this the data for the phenol degradation. We had I have taken that research paper, one of the research paper which used the steel industry wastewater for degradation of the phenol. The growth kinetics, it is the growth kinetics. It is the comparison of the COD removal. COD removal was found out with and without addition of mineral salt treatment, that is during the culturing. So 3 ways it was cultured, that is the without the salt addition and with the salt addition, again wastewater plus mineral, here wastewater plus double amount of the mineral, means increasing the amount of the mineral also. So here it is compared, and you can see that when double amount of mineral is added, that is green color, it is the COD removal degradation is more. Here the correlation was developed for the COD removal against the system parameters, where the parameters were bed height, where bed height was varied flow rate of the gas, flow rate of the water, then the retention time, time of the that is how much time the it was allowed to grow, then the V B by V R ratio. With this correlation was developed. This is the correlation. So, it was found that pH level for the steel industry effluent after the microbial treatment is reduces from the 7.3 to 8.6, which are near to neutral range of the pH of water. Highly efficient performance of the inverse fluidized bed bioreactor stems from the high biomass connection, which is enhanced or which is achieved due to the immobilization of the cells onto or into the solid particles. Now, it is found that also after 3 weeks, means because biomass loading practically constant for the period of 3 weeks. After 3 weeks, it is visible that is the biomass growth is there, means by seeing that solid particle also we can see, and the bioreactor performance is satisfactory, and that is with approximately 70% COD removal, and good performance of the bioreactor was achieved. So, this equipment well suited for the treatment of the industrial wastewater, including samples containing phenols. So, COD removal was found to be 84% with the continuous culturing mode, and therefore, inverse fluidized bed can be used as an effective bioreactor to treat all types of the industrial effluents biologically. This was the conclusion for that research paper, where the Tanmay Limma and myself were published. Tanmay Limma, one of the MTech student, were there. Then phosphate ion also. Here for the phosphate ion, it was also same inoculum was used from the biological that unit from the steel plant industry. We are collected the sludge from there. With that we that is microbes were cultured, and here it was phosphate sources. If you see, wastewater stems from the household streams from the household and the industries contain significant amount of the phosphates. Major source of phosphates in these streams are human excreta, widespread usage of detergents, cleaning agents, and laundry services, and waste containing food and eatable as well. So, besides these sources, industries which deal with wide variety of chemicals also discharge phosphate enriched wastewater. So, our municipal water contains municipality water contains lot of phosphates, because we are that detergents and the here the cleaning agents were being used, and that is drained out, that is also containing lot of, because of that lot of phosphate ions are present. And also sometimes water runoff from the agricultural lands using the fertilizers majorly leads to the phosphates entering the wastewater stream also. These phosphates are mostly present in the two forms, dissolved form or the particulate form, and dissolved form mostly in the orthophosphate form, and some of the phosphates are also present in the polyphosphate form. So, excessive discharge of this phosphate into the water bodies leads to eutrophications. So, here thus because of this what happens, there will be uncontrolled growth of algae and cyanobacteria, which will block the sunlight from reaching the lower levels of the water and causing the plants to die. When the plants will be dying because of the growth of this algae, the uncontrolled growth of algae and cyanobacteria. So, there is also some reduce the oxygen content of the water level, and also the it will cause the death of, when the oxygen content of the water will reduces. So, naturally aquatic life will be affected, and thus algal blooms resulting from this phosphate concentration will be toxic and makes the water body also unfit for the utilization. So, here this was the growth kinetics for the microorganisms. the which was used for the phosphate reductions. So, this was the ESM plot which ensured that there is the film formations on the surface of the particles, and variation in the phosphate reductions concentration. So, a sample plot, because it was 10-12 experiments were there. So, it is found that concentration of the phosphate is gradually decreasing with the time. So, this is also for the different initial concentrations it was compared, all with the all initial concentration phosphate reduction is found to decrease also. Now, you can say removal of the phosphate wastewater. So, here similarly conclusions, here also we can same conclusions. It is as phosphate reduction is possible. So, it can be used as a bioreactor for the treatment of the wastewater. Similarly, for the you can see for the sulfate ions, you can see here sulfate ions, here the growth kinetics, here also is same plot, and you can see with the time how the concentration is reducing. Here you can see initial sulfate concentration, fine sulfate concentration of water. Here you can see variation in the for the different initial concentrations are with the sulfate ion decreasing, and here the air flow rate, and here the pH how it is affected, pH is changing. So, this is again another example for the ammoniacal nitrogen. Here also we can see the that hydrodynamics, we can see solid hold up versus UG, and this is the solid hold up for the VB by VR ratio. This is the liquid hold up for the gas velocity, and you can see growth kinetics for the microbes. Here the ESM analysis. Thus, here also it is found to how ammoniacal nitrogen concentration is reducing with the time. So, it is for the 3 different initial concentrations you can see, and this is the efficiency, how you can see how the efficiency with the time also changing, that is ammoniacal nitrogen reduction is changing, means increasing with the time also. So, thus we can conclude that inverse fluidized bed bioreactor can be operated continuously as under semi batch mode as a stable performance which gives stable performance for the 27 days. So, we can conclude that it is most suitable bioreactor for the treatment of the industrial wastewater. So, thank you.